All right, guys, this is the protein synthesis practice video. Let me get this. There we go. Okay. Protein synthesis practice. Real quick, remember, protein synthesis means to make proteins. And what is a protein? A protein is a chain of amino acids. So we need to back up a little bit and we need to go into the cell. And I have a little drawing here for you. Here's the cell. That's the cell membrane, the cytoplasm. You find that large organelle, the nucleus, which contains our DNA. DNA has our genetic information. In the cytoplasm are organelles called ribosomes. Ribosomes are the site of protein synthesis. So that's where proteins are assembled and made. So how does the information get from here to here? That's the mystery that needed to be solved during this with the central dogma. The central dogma states that information flows from DNA to RNA to proteins, and then proteins code for a trait. So what does this look like? How does this happen? All right, first things first, we have our DNA. The DNA is going to read ATA, adenine, thymine, adenine, GCC, guanine, cytosine, cytosine, CAT, cytosine, adenine, thymine. That's the information that's trapped in there on the DNA. We spoke of transcription. The first step of protein synthesis is transcription, where that information from the DNA gets copied onto a messenger RNA. So when transcription gets copied onto a messenger RNA, A is going to turn into a U, T is going to turn into an A is going to turn into a U, G goes to C goes to G, C goes to G, C goes to G, A goes to U, T goes to A. Remember, in RNA, uracil replaces thymine. This messenger RNA is single-stranded. It can sneak out of the nucleus and go find itself a ribosome. And when it finds a ribosome, the anticodon carried on the ribosome is going to pair up with the codon on the messenger RNA. So for the transfer RNA, U is going to match up with an A is going to match up with a U is going to match up with an A, C goes to G, G, C, 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 A, and U. Here's the important thing, the most important thing you guys need to do right now. Before we take our next step, before we figure out which specific amino acid our transfer RNA is bringing with it, before we do that, we need to cross this off and forget about it. The anticodon is important in getting the amino acid here, but it's not important for our next step. So we need to make super sure that we are just forgetting this for now. We don't need this now, because now we need to figure out which amino acid sequence is represented with this code on the messenger RNA. So even though this seems confusing, it's just the way that it is. We're reading the code from the messenger RNA, not the transfer RNA. We've been talking a lot about a code, and when you have a code, you need a special decoder. The decoders that help us figure out which amino acid codes for a specific sequence on the messenger RNA are called amino acid codon decoders. So you can just Google that and look it up. There's this format, the first base, second base, third base format, and then there's the ring. You have a preference, whichever one's easier for you. I'm going to show you how to do it both ways. So first way, using the first, second, third base positions for the codons. So we need to decode the messenger RNA to figure out which amino acid it codes for. So taking a look at UAU, go to our decoder. U is in the first position, so it means it's somewhere in this row. A is in the second position, so it's somewhere here. U, A, U. U, A, U. The amino acid that codes for UAU is tyrosine, T-Y-R. So that's this first little ball that's dropped off by the tRNA, tyrosine. The next one, we move down the strand of messenger RNA, transfer RNA anticodon comes over, drops off its amino acid. Which amino acid does it drop off? Let's take a look. C means it's somewhere in here. And this is what we're looking at. C, G. Okay, so we're somewhere in here. G. So arginine, A-R-G. Great. That's this one. Okay, which is the next one that gets dropped off? We're looking at GUA. First base, G, so it's somewhere here. U, so it's somewhere here. A. GUA is valine. GUA, valine. Now, you are not going to need to memorize this. This is going to be provided for you. You can look it up, but you will need to be able to figure out the amino acid sequence based off of the strand of DNA. Let's look at another one, and I'm going to show you the other way to do it. So first of all, I'm going to keep our picture out here so we can still see that. Okay. Again, the DNA is found in the nucleus, trapped in there. It's got to transcribe its information onto the messenger RNA. The messenger RNA holds the codons. The most important thing to do is ignore your transfer RNA, cross that out so we don't accidentally look at it, but that's the anticodon. I didn't write that. Okay. So C turns into a G, C turns into a G, C turns into a G. A to U, T to A, G to C, G to C, C to G, A to U. That's our messenger RNA strand. This is the information that we use to figure out which amino acid comes over. Let's look at the circular disc version of this. Some people prefer this, other people prefer this. I don't mind which one you use. Okay, so for this one, you start in the middle. This is position 1. So G, G, G. That means this amino acid, the GGG, codes for the amino acid glycine, GLY. Let's look and see. Yep, glycine. U, A, C. We go U, A, C, and we get tyrosine, T-Y-R, tyrosine. C, G, U, C, G, I have to look, U, back to arginine, ARG, A-R-G. Correct. All right. Let's look at another one. All right, so for page 3 here, I've given you the same DNA sequence as I just did, but I've created a mutation. So instead of ATG, oopsie, a mutation slipped through and it reads ATT. So everything else remains the same. So this is a point mutation. Everything else remained the same. Let's decode and see how this reflects in the amino acid sequence. So I'm taking this away. We're going to look at this one. All right, again, C goes to G, got it. A goes to U, T goes to A, T goes to A. G goes to C, C goes to G, A goes to U. All right, now we figure out what the transfer RNA is, but then we don't pay it no mind. We just ignore U. G, G, G. G, 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 glycine. I've got glycine. UAA. UAA. I've got a stop. Excuse me. A stop codon. So we're gonna write down stop here. And I'm just gonna finish it out, even though it wouldn't actually finish out. C, G, U, arginine. Okay. So what does this mean? First of all, the stop codon means we're going to stop going through the process of protein synthesis, and this amino acid chain is going to break off and be shorter than it's supposed to be. This is called what? Do you remember this from your vocab or from our presentation? This is called a nonsense mutation, and this is what occurs when the amino acid sequence ends up being shortened because of a mutation. Okay, so that's how that can result. Let's take another look. We have DNA sequence, TAA, GTA, CCG. The messenger RNA, T goes to A goes to U, A goes to U, G goes to C, T goes to A goes to U, C goes to G, C goes to G, G goes to C. We can figure out what the transfer RNA is, but then we just ignore it, and then we're going to figure out the amino acid sequence. Where'd my paper go? All right. AUU. AUU gives you ILE, which is isoleucine. CAU. CAU gives us histidine. Great. GGC. GGC gives us glycine. Now, you may not think it's important to actually cross out the transfer RNA, but I recommend it. Like, I physically have to cross it out so I don't accidentally decode off of the transfer RNA by mistake. All right, let's take another look here. We have another mutation. Oh no. All right, so we have the same sequence as last time. Last time we had TAA, GTA, CCG. The mutation is right here. Instead of being CCG, it's CCC. So we're going to go through, come up with AAU, CAU, but now this time we end up with GGG. Let's take a look and see how this reflects on our amino acid sequence. AAU, AAU, wait, AUU. AUU is going to give us isoleucine. Yep. CAU is going to give us histidine. Yep. And GGG. This is where we have our mutation, but it gives us glycine. Hold up. We got glycine last time. Okay, so what this shows us is that GGG and GGC both code for the amino acid glycine. So what does this tell us about this mutation? This is a silent mutation. It's a mutation where the amino acid sequence is going to remain the same, so you're not going to see an impact on the phenotype of the organism. All right, guys, you've listened to me enough. On a separate sheet or on a doc whatever you need to do. I would recommend writing it. I think it's very hard to, I guess you could type it, whatever makes you happy. The DNA sequence is A, G, C, T, A, G, C, A, T. So what you need to do first is your transcription. You need to figure out what your messenger RNA is going to say. Then you need to do your translation for your transfer RNA, and then you're going to cross that out. And then you find out which amino acids you have using an amino acid codon chart. An amino acid codon chart, whichever type you prefer. You're going to give me the 3 amino acids that result from this strand of DNA. Do remember the rules for base pairing. A goes with T, G goes with C, unless your RNA. No T's in RNA. We use a U instead. No T. Looks like I wrote no tin. No T's in RNA. We use a U instead. So pause the video and go through this sequence. When you come back, we have, when we come back, we have another for you to take a look at. This is your try it on your own number 2. So pause the video here and go through this one and find the amino acid sequence. All right, I'll give you the answers on another link.